Dentinogenesis is the process of formation of dentin, and its formation is carried out by cells called odontoblasts. Dentin is the calcified tissue which forms the bulk of the tooth, whereas the odontoblasts are present in the pulp and their processes extend through the thickness of dentin called odontoblastic processes. If we take a look at the chronology of teeth, formation of primary dentin begins just before the beginning of enamel formation. Then it continues even after tooth eruption and ends with root completion. However, now the secondary dentin formation begins, which continues during the whole lifespan of the tooth and ends only when the tooth dies. Differentiation of odontoblasts. The dentin of a tooth can be divided into two parts, dentin in the crown called coronal dentin and dentin in the root, the rooty dentin. Let us first see how odontoblasts of the coronal dentin are formed. During tooth development in the enamel organ, the cells of the inner enamel epithelium are separated from the cells of the dental papilla by a cell-free zone. The cells of the inner enamel epithelium then transform into ameloblasts. During the life cycle of ameloblasts, as the cells enter the organizing stage, they become tall columnar. This eliminates the cell-free zone and the ameloblasts now come in direct contact with the cells of the dental papilla. The ameloblasts then release signaling molecules and growth factors, because of which the undifferentiated cells of the dental papilla differentiate into odontoblasts. Odontoblasts are tall columnar cells, around 7 microns in diameter, 40 microns in length, and with nucleus positioned towards the dental papilla. Now the differentiation of root dentin odontoblasts. This also occurs during tooth development, but at a later stage after the formation of Hertwig's epithelial root sheath. Hertwig's epithelial root sheath is a bilaminar layer made of inner enamel epithelial cells and outer enamel epithelial cells. It is this sheath which releases signaling molecules and growth factors to the undifferentiated cells of the dental papilla and differentiate them into odontoblasts. Formation of odontoblastic processes. This is a unique feature of dentinogenesis. As odontoblasts deposit dentin and move backwards, they leave an extension of cells in the forming dentin. As cells continue moving backwards, these extensions continue growing in length. These cell extensions are odontoblastic processes, which extend from the odontoblasts to the dentino-enamel junction. So till now we have studied about the formation of odontoblasts and formation of odontoblastic processes. Let us now study about the formation of the calcified part of dentin. Dentinogenesis occurs in two steps. Step 1, formation of organic matrix. Step 2, mineralization. First, the formation of organic matrix. The organic matrix is made of proteins, of which collagen forms 90%. In this collagen, majority is formed by type 1 collagen and small amount by type 3 collagen and type 5 collagen. In non-collagenous proteins, there are dentin phosphoprotein, dentin siloprotein, dentin glycoprotein, dentin matrix protein. The odontoblasts release all these proteins into the stroma. A scaffold is formed by collagen type 1 which accommodates large portion of minerals, whereas the non-collagenous proteins regulate this mineral deposition. They may act as an inhibitor or promoter of mineralization. Pre-dentine. As odontoblasts start formation of dentin, the first layer that is deposited, the layer nearest to the odontoblast, is made only of organic matter called pre-dentine. It has no mineral deposition as yet. It is similar to the organic layers seen in other mineralized tissues. So as dentinogenesis goes on, this pre-dentine layer will always be present, no matter if it's the primary dentine formation, secondary dentine formation or tertiary dentine formation. The zone from which mineralization starts is called the mineralization front. It is because of the fact that mineralization always lags behind organic matter formation. Mineralization. The main inorganic portion of dentin is formed by the hydroxyapatite crystals. They are about 3 nanometer wide and 100 nanometer long. These crystals are larger than the ones present in bone, but way smaller than the one in enamel. Mineralization mainly occurs by matrix vesicles, in which Odontoblasts release membrane-bound vesicles into the organic matrix. They are 25 nanometer to 250 nanometer in size. They contain hydroxyapatite crystals, calcium ions, phospholipids, and many such chemicals which initiate and promote mineralization. There are two patterns of mineralization: globular or chalcospheric pattern and linear pattern. Globular pattern is mostly seen in the coronal dentine near mantle dentine, linear pattern more in root dentine and near the pulp. The rate of mineralization is faster in globular pattern than in linear pattern. In globular mineralization, the odontoblasts deposit the hydroxyapatite crystals in several areas of the collagen matrix. These crystals then grow centrifugally with deposition of more minerals. Eventually, these enlarging globules fuse to form a single mineralized mass. In case there is hindrance in the fusion of these globules, the produced hypocalcified areas are called interglobular dentine. In linear mineralization, the crystals get deposited along the collagen fibers. These crystals fuse together early at a smaller size and so make the mineralization front appear in a straight line. This was the process of dentinogenesis in primary dentine, which is same in secondary dentine and in tertiary dentine. The difference is only in the rate of dentinogenesis. Secondary dentine formation is much slower compared to primary dentine deposition. However, rate of tertiary dentine formation is inconsistently rapid, due to which irregularities are commonly seen in its structure.